Up to now, we have discussed about the RMP and action potential. With a very brief review of the previous lectures, in RMP, resting membrane potential, there is simply there is no conduction system, there is no, you can say, the responsiveness to the stimulus. And during the RMP, resting membrane potential, cell is always permeable to potassium and cell is internally electronegative. And whenever you stimulate the particular cell, which is always the neuron or muscle cell, remember, beside to these cells, and there is no any cell which is excitable cell throughout the kingdom Animalia. Whenever you stimulate the cell and you are success to achieve or take the RMP up to the threshold value, then suddenly the sodium channels will on and the sodium will come inside to neutralize or depolarize the cell membrane up to the positive level. And when it reaches this maximum level, like near to the equilibrium potassium for the sodium, suddenly the sodium channels will off and potassium gated channel will open and potassium will rush outside the cell, bringing cell membrane again its resting level, repolarize the state, and due to the delay of the time, the little channels will open, so the potassium will keep moving outside, so the cell become hyperpolarized. But after that time, the potassium channels will completely off and it will become recovered to the RMP, resting membrane potential. This is what we have learned, and we have also seen the propagation of the nerve impulse, how when one segment is stimulated, the sodium trickling inside, it will depolarize the adjacent segment, and then this wave of depolarization, which is followed by the wave of repolarization, spread over all the neuron. Ultimately, it reaches to the synaptic terminal. Remember one more thing. Actually, there is a neuron to neuron talk, but there is also neuron to muscle talk. Whenever neuron to muscle talk, this junction is known as neuromuscular junction, which we will discuss in later lectures, not right now, because this is not the part of FSc medical, even not this is present in the OA levels, but we are not discussing right now. Our main focus is on the FSc level. So today we will see, because the major things like RMP and the action potential we have seen. Now the very basic things which we have discussed is about synapses. Synapses mean the two membranes come together, or the two things come near to each other and to talk to each other. Now synapses, first of all, very basic information. Synapses simply is the neuronal talk, or the membranes come together and exchanging the information. This is known as synapses. Synapses may be two types. One is, we can say, this is a chemical synapses, and then we call it electrical synapses. The more common synapses found human body is chemical synapses. It doesn't mean that the electrical synapses is not present. Amacrine cells of the retina are also doing the electrical synapses, but we will discuss the properties of the two, both chemical and electrical. Now listen. I have made two hypothetically neurons which are arranged in a parallel fashion. Suppose action potential we stimulate here, and the action potentials propagated on the membrane of the cell membrane of the neuron, and ultimately depolarize the terminal knobs. Actually, you must be knowing this is a cell body, this is a zone, and these are the terminal knobs. These are the terminal knobs. This is clearly seen in video. These are the terminal knobs. In the terminal knobs, there is a vesicles. These are the vesicles. These vesicles are formed either cell body or either at the axon. But the enzymes which are utilized to make the vesicles and neurotransmitters are always present in the cell bodies and then travel along the axon. In these vesicles, there is a neurotransmitters. There is a neurotransmitter present. These are the chemicals that neurons release. And the chemical that neurons release, these are the neuronal transmitter, or we can say simply these are the neurotransmitters. So these are the vesicles. These are the vesicles, and inside vesicle there are thousands of the neurotransmitters, and now if just suppose this is neuron number 1 and this is neuron number 2, and the action potential comes from neuron 1 and the action potential comes from the neuron 1, then this neuron designated as this is the presynaptic neuron. This is the presynaptic neuron, and this knob will determine or designated as presynaptic knob. This knob is designated as presynaptic knob because this is later to the this neuron, adjacent to the this neuron. This neuron is designated as postsynaptic neuron, and this cell body or this membrane, okay, this is the membrane, presynaptic membrane, or we can say knob, presynaptic membrane, and this is the membrane of postsynaptic neuron. So we call this membrane as the postsynaptic membrane. These are the definitions just simply. And the space between presynaptic neuron or presynaptic membrane to the postsynaptic membrane, this is a very minor space which is looking huge in my diagram, but is not so much huge. This is a very minor space, and this space is known as synaptic cleft. Am I clear? Now if the action potential come from this side, so this is the presynaptic neuron or this will be the postsynaptic, but in my diagram in a particular example action potential comes from this direction, so this membrane will become the presynaptic membrane and this will become postsynaptic membrane, and the space between the presynaptic and postsynaptic, this is the synaptic cleft. Such typical arrangement of the neuron in which one neuron and then the synaptic cleft and then there is a postsynaptic neuron, this such type of synapses is chemical synapses because there is a chemicals involved. Remember, these are the chemicals involved in such kind of synapses, so we call it chemical synapses. The main property of the chemical synapses is this is the one direction, unidirectional. This is relatively, you can say, slow as compared to electrical. As compared to the electrical, it is slow, otherwise it is also not the slow. So relatively electrical synapses, it is slow, isn't it? Now electrical synapses, what electrical synapses? In electrical synapses, actually if these are the neuron and this is the another like this, there is a gap junctions, between the adjacent neurons. There is no synaptic cleft, no neurotransmitter, simply the propagated nerve impulse move from these windows, these intercalated discs, these gap junctions, and similar electrical synapses you can see in the cardiac cells, because in cardiac cells all the myocardial cells connected with each other with the intercalated disc. So this will propagate through these gap junctions and depolarizes the adjacent neuron. So these are the electrical synapses, and the main property it is the bidirectional. Like nerve impulse can move this side, also can move this side at the same time. Understand? This is the little difference between the electrical synapses and this is the fast. This one is the fast. But our main concentration is on chemical synapses because at FSc level there is no electrical synapses given in the book, only chemical synapses present. In chemical synapses, what really doing that the electric nerve impulse which have we seen previously propagated along the cell membrane of the neuron ultimately reaches to this point, and then we have discussed this point, the calcium channel will open, calcium will come inside and fuse with this, and then ultimately bring the vesicle near to the synaptic membrane, and then this will fuse with the synaptic membrane, and then neurotransmitter will release in the synaptic cleft which will stimulate these neurotransmitters receptor for these neurotransmitters, which are the proteins, are present in the postsynaptic membrane. Remember, now there is a very important concentration. These neurotransmitters, the widely accepted hypothesis was that the one neuron contain only one kind of neurotransmitter. First of all, I would like to tell the neurotransmitter classification. Neurotransmitters. The mashhoor zamana neurotransmitter is acetylcholine. Acetylcholine. This is a class 1. First discovered neurotransmitter is acetylcholine. Then there is amino acid also, like glycine, like you can say the aspartate, glutamic acid. Then biogenic amines also present, like GABA, gamma amino butyric acid, and then there is also dopamine, serotonin, and no doubt another mashhoor one after acetylcholine, which is norepinephrine, which is also known as noradrenaline, and epinephrine also, but epinephrine comes from the circulation from the medulla of the adrenal gland, epinephrine. So these are the neurotransmitter. The widely accepted notion in the previously is that one neuron contain only one kind of neurotransmitter, but the ample evidence is nowadays is suggested that the one neuron can contain multiple neurotransmitters. But in one vesicles only one kind of neurotransmitter is present, and at a particular time only one kind of neurotransmitter is released depending on the frequency and the aptitude of the neuron and frequency of the action potential. So this is about the neurotransmitter. Now these neurotransmitters determine that either the adjacent or next neuron should be excited or should be inhibited. Actually, you know, this is a very simple. Whenever you feel someone touches you hard, you feel the pain. Actually, your neuron is highly excited. That's why you feel the pain. But sometimes, during the tooth cavity filling or during the high injury, if someone inject you or you can say local anesthesia, you will numbed. You don't feel any pain. Why? Because the neurons at particular site is inhibited. So that's why you cannot sense the pain. So these neurotransmitters determine either the next neuron should be inhibited or should be excited. So those neurotransmitter which has excited nature to the next neuron, so we call it, there is the excitatory post synaptic potential, we call it. If this, just suppose this neurotransmitter excite this neuron mean it will become positive inside or negative inside. If the neuron I say excited, it mean the neurons internally positive simply. So this is, we can say, excitatory post synaptic potential, this neurotransmitter produced in this cell. Just suppose this neurotransmitter release and it will make this neuron more hyperpolarized, mean inhibited. So this cell will become more electronegative. So we can say this neurotransmitter has a nature of the potential which is inhibitory post synaptic potential. This is little bit explanation of the how neuron talk to each other. That, remember, in one vesicle there is one kind of neurotransmitter. These neurotransmitter may excite the other neuron or may inhibit the other neuron. How they excite? Like there is of course, if you have the very good concept about the action potential or the resting potential, you can interpret with your common sense that if the neurotransmitter release and I have to inhibit this neuron, so what does this neuron bring the changes in this neuron? It will bring the changes as that it will open the potassium channels, so potassium will move out, and it will open the chlorine channels, that chlorine come inside, so this cell will become still more electronegative, more hyperpolarized, more inhibited. Opposite to that, if the particular neurotransmitter wants to become this cell more excitable, so what are these neurotransmitters bring the changes in this membrane? Of course, they will shut off the potassium channels, they will open the sodium channel drastically, so the sodium will come inside and cell will become internally more positive. So we can say cell is become excited. One thing, the neurotransmitter actually by action neurotransmitters has two kinds of action, either direct or either indirect. What does it mean, direct or indirect? Direct means if the neurotransmitter attaches the particular receptor and this receptor will open simply, like neurotransmitter itself opens the channel. So this why this is the direct action. But sometimes this is not the case, sometimes this is the indirect, and I am not going to detail about this. This is actually the G coupled receptors, G stimulatory, G inhibitory system present over there, that the second messenger system. This is the indirect action, this is second messenger system, that the neurotransmitter comes inside, comes to the next neuron, stimulate the receptor, and this receptor makes the changes in the second neuron, bringing the second messenger, and then it will phosphorylate the channels, and then the channels will open depending on the conditions, either the positive channels or either the negative channels. Like positive channel, negative channels mean either the chlorine channels will open, that take the chlorine inside, so the neuron inhibited, or either take the potassium outside, so again the cell inhibited, or making the sodium channel open, so the cell become more excited. So this is all about the synapses. That synapses may be the chemical synapse or the electrical synapse. This is the terminal talking. This is the propagation of the nerve impulse. Now one more thing I have skipped in the previous lecture. Actually this propagation, now this is the velocity. We can enhance or speed up the velocity of the nerve impulse, this impulse. Actually in my previous video you can find on my YouTube channel, that is about the myelinated neurons. You know, if this is a neuron one and this is another neuron, if this neuron is myelinated, this neuron is myelinated by Schwann cells, particular regions, you know, myelin is lipid layer, so it can insulate these regions. So do you think the nerve impulse move fastly around this or this? Actually nerve impulse moves very speedily in myelinated neurons. Why? The nerve impulse come this, then jump to this, then jump to this, then jump to this. Actually in myelinated neurons, nerve impulse only moves from nodes of Ranvier, nodes to nodes. But in this case it has to travel all the pathway. So this is slow conduction and this is fast conduction. Understand? One way to speed up the nerve impulse is something like this. Another way, if you dilate, like some neurons have wider diameter. If the neurons have wider diameter, then of course there is a less resistance to the electrical charges, so the electrical current moves fast. So one is the diameter large, conductivity large, and then is a myelination. If the myelinated neurons present, the conductivity enhanced. One more thing, the conductance of the nerve impulse is more high in the neurons of warm blooded animals rather than the cold blooded. So this is the high conductance in warm blooded. And don't forget, we are warm blooded or cold blooded? You are cold, but I am warm blooded. Understand? This is we are warm blooded animals, isn't it? So these are the conductance of the nerve impulse. Actually now one more thing actually, just suppose this is the I am giving the concept of the myelination. You will tell me the myelination. Just suppose, actually if I cut like this section, it will looks like this. This is your axon and this is your Schwann cell, isn't it? Again, like this will become like this. Okay, now tell me which neuron is myelinated, which is not myelinated? 1, 2 or 3? You said the second one is myelinated. Actually, the answer is wrong. Now listen. Number 3 is myelinated. Number 2 is non-myelinated. Number 1 is diseased. What I want to put in your mind? I think recording is not well going on. Actually, all the time Schwann cells are present with the neurons. All the time Schwann cells present with the neurons. If they make the multiple layers, then we say this is the myelinated neurons. In this case, only Schwann cell present to the neuron. There is no multiple layers. We can say this is non-myelinated. But some diseases has disappeared, disintegrated Schwann cells. So these axons are diseased. So these are the little concept about the myelination and the conductivity of the nerve impulse and the synapses. So in later lectures we will discuss more about the neurology. This is all about today. Thank you.